This is Chris with the Ancient Scholar. I hope you guys are all doing well, and be adding another video to the playlist of the Biology 225 Human Anatomy and Physiology 1 playlist. If you are not one of my students in this class, the videos are going to kind of pop up and seem really out of place simply because I'm not following a real coherent structure. I'm just doing a whole bunch of videos in anticipation for areas of this course where people may have more difficulty understanding stuff. And at some point in the future, I'll go ahead and I'll arrange the playlist so it's a bit more coherent and the flow makes a little more sense. But I'm just doing a bunch of videos right now, and then I'll be linking those videos into my into the Canvas for my students. So everybody gets to benefit from these videos. They'll just kind of seem out of place if you're one of my YouTube viewers and you're going to say, well, why did this pop up all of a sudden when you didn't talk about this? And that's why I'm not really doing these videos in any specific order. So what I'm going to be doing today is I'm going to be talking about amino acids and proteins. And as we've talked about earlier, even though it doesn't seem like it necessarily, but as we talked about, there are what we call the macromolecules of life, and these are the significant molecules that are most important in the day-to-day biochemistry encountered in most living organisms. And these molecules can really fall into four general classes, and those general classes, just to recap, are the carbohydrates. Right, the carbohydrates, okay, the fats. The fats are the fatty acids. All right, proteins, and finally nucleic acids. All right. Now, carbohydrates, of course, are very important as far as energy production goes, and a whole bunch of other concepts that we'll talk about. Fats are very important. They are a critical component of the cell membrane. Fats are also an important part of a certain steroid hormones, and a very important part of giving some fluidity and additional structure to the cell wall in the form of cholesterol. Proteins are critically important when it comes to virtually every kind of biochemical process in the human body, be that a channel that allows ions to flow in and out of the cell, an enzyme that helps make chemical reactions occur in the body more efficiently, be it a receptor. It could be a receptor for a certain hormone or a certain chemical or stimulus or drug. And then, of course, proteins are very important when it comes to certain tissues, specifically contractile tissues like muscle tissue. And you even have proteins that help move stuff around the cells. Proteins like kinesin, which is literally kind of walks along the cytoskeleton, moving stuff through the cell. So proteins are also important as far as what cells use to identify self from non-self. When you talk about the immune system, they're an important part of blood clotting and clotting factors, and so on and so forth. Just critically important. Proteins really are the things that get the day-to-day processes within our body going and keep them going. And then finally, we have nucleic acids, and nucleic acids, of course, are kind of like the blueprints of the cell and the instructions to tell the cell how to make the proteins, these proteins that get all of this stuff done. That information, of course, is contained within nucleic acids. So what we're going to be talking about today is we're going to be talking about the proteins. So what do we need to know about proteins? Well, proteins actually come from amino acids. So amino acids are the fundamental molecules, the fundamental molecular building blocks for proteins. And when we talk about human anatomy and physiology, what we're talking about is there are about more or less 20 different amino acids 20 amino acids that the human needs, and various combinations of these 20 amino acids are what ultimately make up all of the proteins in the human body. And then, of course, there are some modifications. You can add what's called a prosthetic group, where you have the protein and you add another non-amino acid kind of molecule in there. Sometimes it's a metal like iron is added. When you talk about a protein like hemoglobin to help transport oxygen, you do have some specialized, what are known as prosthetic groups, but the bulk of the protein structure comes from these different combinations of these 20 amino acids. And in the human, you have two general types of amino acids. You have non-essential. Okay, you have non-essential, and then you have essential. All right, essential amino acids. Non-essential amino acids are amino acids that the human body can produce. Okay, so the human body could actually produce these amino acids if it needs to. However, the essential amino acids cannot be produced, and the only possible way for the human, at least, to get these amino acids is to consume them. Okay, consume them. So we have to eat foods that have these essential amino acids, or we will not be able to make many of the proteins that we need to make. So of these 20 amino acids, there are 9. We generally say there are 9 amino acids that are essential. All right, and I'm not going to necessarily make you memorize all 20 amino acids. You may have to do that in other classes. This is more big picture, but the 9 essential amino acids, I'll just kind of go through them, include the following, and I won't write them down, I'll just kind of throw them at you. They include phenylalanine, valine, threonine, tryptophan, methionine, leucine, isoleucine, lysine, and histidine. And many of these amino acids, these essential amino acids, are critical when it comes to very important molecules such as certain neurotransmitters, for example, and very important enzymes as well use these essential amino acids. So these truly are essential. The only way to get them is through your diet, through foods that are high in these. So certain types of beans, for example, that may be high in these essential amino acids. Otherwise, we can also eat meat. Okay, if we eat meat, meat, of course, is muscle, basically. For the most part, it's muscle from an animal. It's cooked, you consume that, and of course, muscle is made up of primarily of protein molecules, and then the body, through digestion, okay, we digest, and there are special enzymes in the human body, the special enzymes that the human body produces, called proteases. Okay, called proteases, and whenever you see a molecule and it ends with ASE, there's a reasonable bet that we're talking about an enzyme. So protease would be an enzyme that would break down proteins. Amylase, okay, amylase would be an amino acid that would break down certain types of carbohydrates. Lipases, all right, again, are enzymes that break down certain types of lipids. So that's kind of a helpful little hint there as well. So the protease enzymes help break down this muscle, break down the proteins, okay, are broken down into their individual amino acids, okay, and then the amino acids can be absorbed, okay, in the small intestines primarily, and then the human body can take those amino acids and turn them into the proteins that the human body needs, that the cells need. So that's what, when we consume meat. Otherwise, the human body can produce non-essential amino acids, and then those amino acids can be used to make proteins. Okay, so there we go. There's kind of the big picture. So let's just go ahead and look at the general, okay, the general structure of an amino acid, and hopefully it'll become apparent why we call them amino acids. So the general structure of amino acid is as follows. Amino acid contains 3 different components. Okay, the first component on one end, it contains a nitrogen, okay, that has 2 hydrogens, okay, covalently bound to it. All right, and then you have this carbon covalently bound to a hydrogen in the middle, and then this carbon is attached to some sort of chemical group, and we call that group an R group, and R just means everything. I just think of it as everything else. This is often referred to as the side chain, and I'll give you some examples here in a little bit. But this is a side chain, and it could be a methyl group or a complex hydrocarbon chain, may even be like a benzene ring or aromatic. It can be many, many different things. And these side chains are very important, but we'll just kind of keep it there. So whenever I talk about R in reference to amino acids and proteins, that just means side chain, okay? And then the other end of the amino acid is a carboxylic acid, and that is a COOH. All right, and that is a carbon double bonded to an oxygen, double covalent bound, and then a hydroxyl group, COOH. Okay, and of course this can dissociate. The hydrogen can pop off as a proton, and that's what makes this an acid. And carboxylic acids are just very, very common. So let's just label these here. Okay, so right here is my acid component of the amino acid. Okay, right here is my what we call amine. And amine comes from the term ammonia, or NH3. So ammonia is a nitrogen, okay, covalently bound to 3 hydrogen atoms. That's called ammonia. And then what we do is, when you replace one or more of these hydrogens with either get rid of them or replace them with another bond, for example, in an amino acid, one of those 3 covalent bonds is replaced by a covalent bond to the carbon, okay? And when you replace one of the 3, we call that a primary amine. When you replace 2 of the 3 hydrogens, you call that a secondary, and a tertiary when you get rid of all 3 hydrogens. So amino acids are examples of primary amines, but this is the amine, and I just think of ammonia. It is derived from ammonia. It is an ammonia. It starts off as ammonia, and then you replace one of the 3 hydrogens with that covalent bond to the carbon. So you have your amine group and your acid, hence the term amino acid. And then this central carbon here that is attached to a hydrogen in the R group, this central carbon is important because of that R group, and we often will call this carbon here the alpha carbon, okay, because there's a lot of important stuff coming. Okay, so what do we do? So what happens is amino acids are combined together. Okay, amino acids are combined together to produce. Oops, get rid of this here. Amino acids are combined together to produce proteins. So let's talk about how amino acids are combined, since that's the whole point of this. So what I'm going to do is I'm going to give you a couple of examples. I'm going to give you an example of amino acid called alanine. All right. All right. So I have alanine here, and so this is going to be in my NH2 group here. All right, my alpha carbon, hydrogen, and then alanine has a methyl group, CH3, and this methyl group, this particular group, is an example of a non-polar side chain, or non-polar R. So this is going to be, because it's not polar, it does not like water very much, and so this is going to be hydrophobic. Okay, so in the aqueous environment, the water environment of our body, this is going to want to get away from the water, and you can start to see that because it wants to get away from water, it may push or pull this particular part of the protein. If this is one amino acid that makes up a protein, it may push or pull and tug it and move it in a certain way because of it being hydrophobic. Now, other types of side chains may be charged or more hydrophilic. They may be more attracted to the water, so they're going to pull in a different direction. And you can see how this sets us up for a situation is as we start adding on more and more of these amino acids to make what's called a polypeptide, and eventually a protein, there's going to be this pushing and pulling and moving around due to the intermolecular forces that these side chains experience. Okay, so, and then we have our carboxylic acid here, COOH. All right, so what I'm going to do is I'm going to take the amino acid alanine here, and I'm going to put it, let's say we're making a protein, and I'm going to have threonine here over here. All right, I'm going to have the nitrogen here, hydrogen here, alpha carbon, hydrogen, and then I'm going to have a CH group, and then a hydroxyl group, and then a CH3, a methyl group. And this hydroxyl, or this alcohol group here, makes this side chain much more polar. All right, and anytime you have oxygen and hydrogen together, the electronegativity of the oxygen is pretty much going to ensure that you have some polar. So this area here is much more polar. So this is going to be more hydrophilic, or more water-loving than this side here. All right. So this is an example of a more polar side chain. And some side chains, some R groups, are actually charged. They may be positively or negatively charged, and of course that sets us up for electrostatic interactions between the different R groups. And then I have my COOH. Okay. So to combine these two amino acids, what happens is we have what's known as a dehydration synthesis chemical reaction. A dehydration synthesis. And so dehydration just means dehydrate. So I'm going to dehydrate these two particular amino acids. So I need to pull a water out of there, and I need two hydrogens and an oxygen. And if you look at this hydroxyl group here and this hydrogen, so the hydroxyl group on the carbon and the hydrogen on the nitrogen, that's an OH and an H, right? And OH plus an H can give me H2O. So what I do is I can pull that water out of here, all right? And so if I pull a water out of there, whoops, let's undo that there. There we go. So if I pull a water out of there, okay, that basically opens this up here, all right? And that allows this carbon to become covalently bound to that nitrogen, okay? And this is actually a very solid, this is a very strong chemical bond, and this is known as a peptide bond. And this is I've now made a peptide, and multiple amino acids, when I add multiple amino acids with this dehydration synthesis reaction, I could put another nitrogen, okay, of another amino acid here, and I could do the same thing, and then I'd have 3 amino acids, and then we'd have what's called a polypeptide. And eventually, as I keep adding more and more amino acids, okay, I'm going to go ahead and erase this stuff here, my polypeptide is going to get longer and longer and longer and longer, okay, and then I'm going to have the NH2 on one end of my original amino acid, and then the next amino acid that gets added, I'm going to have the COOH here, and some people call this the N terminus. Okay, that's the N where I have my amine group, and then the C terminus here is where my carbon is, the carboxylic acid. So I'm going to add more and more and more of these, and then what's going to happen is I have all of these R groups here, right? All of the side chains of those amino acids are just kind of hanging out here. But what's going to happen is these R groups, right? These R groups all have carbons, oxygens, hydrogens, different chemical properties to them. And as this thing gets longer, these R groups are going to begin to interact with one another, right? And interact with because this is an aqueous environment. And so what's going to happen is as this polypeptide gets longer and longer, the R's, depending on what amino acids are next to which, are going to start interacting. And what's going to happen is this is going to start folding and twisting around. And so when that happens, the protein begins to take its shape, right? And in a kind of an underlying theme of anatomy and physiology is, of course, structure. All right, structure fits function. And every protein has a special shape, a very specific shape called a conformation. And the conformation of the protein is what allows a protein to function, allows it to be an enzyme or a receptor or some sort of channel, an ion channel or a sodium-potassium pump or what have you. And if I change the shape of the protein, I change its conformation, I can change its function. And sometimes we want to do that, and other times we don't want to do that. But proteins require this special shape. And when this shape is changed in a way that is not good, or a way that prevents the protein from working, there is a term, and this is called denature. All right, well, if I denatured it, or I denature a protein, that is changing the protein shape in a way that prevents it from doing its special job. And this can be done in several different ways. This may be the result of heat. If I heat a protein up, I can denature it. If I add acid, okay, strong acids, strong bases, alcohol, okay, all of these things can do it. Certain toxins can do it. Okay, so if I get really sick and I get, like, let's say I get a really, really high fever, that really high fever may start denaturing and deactivating certain proteins. Or maybe I get really, really cold, I get hypothermic. If I get hypothermic, I'm really cold. Believe it or not, being really cold denatures or changes the shape of your clotting factors and prevents you from clotting adequately. And being really cold can actually make you more prone to bleeding. And sometimes people that um, have had significant traumatic injuries are much more prone to bleed to death when they're cold, um, and so on and so forth. Becoming acidotic, if you're really sick and you develop a metabolic acidosis or really bad respiratory acidosis, your respiratory system isn't working, that can denature proteins and cause lots of problems as well. So certain drugs and toxins as well. So not a good deal. So I guess the next big question is, well, how do these proteins take on the shape, and what levels of shape, quote unquote, do we have? And we say that there are 4 levels. Okay, 4 levels of protein structure. Okay, the primary level, the primary level is going to be the peptide bonding. Okay, so it's going to be the amino acids combining due to the dehydration synthesis resulting in the formation of a peptide bond. Okay, so the primary structure is going to be, to draw this here, I have my N-terminus, I have all the amino acids, and then I have my C, my carboxylic acid here, and then I have all of my R chain, my side groups here. Okay, that's the primary structure, this peptide bonding. Very robust, very difficult to destroy the primary structure of a protein. But then what happens is, as that polypeptide gets longer and longer, these R groups start interacting. And typically what we're going to see is, we're going to see this, if I just simplify this, okay, we're going to see this strand kind of twist and fold itself into 2 classic structures. It generally will do one of 2 things. It will fold itself into what's known as an alpha helix. Okay, so it's going to have a helical kind of shape that it'll twist itself into, or a beta sheet. Okay, where you get this strand kind of pleated, it becomes pleated, or it might fold in on itself like this. And you can even, as you get more and more amino acids, you can even get, you know, this complex structure of both beta pleated sheets, and then maybe a helice here and some more pleating, and then another helix here and some more pleating like that, and you can get that kind of structure here. So the alpha helix or the beta pleated sheet, just these guys right here, is what we call the secondary structure. And the secondary structure is typically the result of hydrogen bonding. Remember, we talked about that in chemistry, one of the most important intermolecular forces, where you have a hydrogen that is typically attached to an electronegative atom, such as an oxygen. So it's attached to an oxygen, a nitrogen, maybe a fluorine, generally oxygen and nitrogen, and so you have a very polar. And so if I have, let's say, a hydrogen that's covalently bound to an oxygen here, okay, you have a separation of charge because the oxygen is more electronegative, and so you kind of have this partial positive on the hydrogen, and so the hydrogen is going to be attracted to something that's negative. So let's say that I have this on one of my R groups, okay, and then on another R group of another amino acid I have, let's see here, maybe like this, an O, and then an H sticking out in this direction, so the H is partially positive, the O is partially negative, and then I have this, right, the partial negative, the partial positive, is going to cause an attraction. It's not a full-on covalent chemical bond, but it's still an attraction, and that's what we call a hydrogen bond, right? Due to the a hydrogen being covalently bound to an electronegative atom, and there's that the separation of charge, this polarity. And so that is what causes the primary part of what causes this alpha helix, this secondary structure, to begin to develop. And then you get additional interactions. You can get what are called disulfide bonds, where you have sulfur atoms. You can get disulfide bonds, kind of look like that. You can get full-on ionic kinds of bonding occur. You can get hydrophobic, hydrophobic and hydrophilic. All right, the hydrophobic R groups may be pushed away from, if this is an aqueous environment and the water's out here, the more hydrophobic parts are going to get pushed in away from the water, and the more hydrophilic groups are going to get pulled out that way. So you have all of these more complex kinds of intermolecular forces occurring, and that is what's going to create our tertiary structure, where you get this combination of alpha helixes and beta pleated sheets occurring. And then finally we can get something called quaternary structure. And that's where maybe I have kind of a protein being produced here, and a protein here, and another protein here, and then these three proteins come together to form one large complex protein. That's what we call quaternary structure, and the classic example of that, of quaternary structure, is hemoglobin. All right, hemoglobin is actually 4 different globulins, all right, that come together to produce the total hemoglobin molecule. You have what are called 2 alpha and 2 beta globins that kind of come together, and each one of them has a little iron-containing area called something known as a porphyrin ring, where the iron is, and the oxygen molecules actually attached to that ring. And so that's where you can get one hemoglobin molecule can bond 4 oxygen molecules, because of this quaternary structure. That's a classic example. And what we often see when we heat something up, when we heat proteins, specifically when it comes to cooking them, when I cook something, when I cook a steak, for example, the steak just doesn't completely disintegrate, right? And so the primary structure of those proteins tends to remain intact. So the peptide bonds that hold amino acids together tend to remain intact because they're a pretty robust bond. And so what happens typically when you heat a protein up is you break the hydrogen bonds, and so you disrupt the secondary structure, and that's what you see when you start cooking meat, for example. Another thing that you can do is you can take an egg. You can crack an egg and stick it in something that has a very high ethanol, like some Everclear or something like that, and the egg will appear to be cooked if you leave it in there. And this is something I'll actually demonstrate in class. And what's happening is that alcohol, right? Those alcohol, because ethanol is just a ethane molecule with an OH, right? And so these OH here, these alcohol groups, are actually disrupting the hydrogen bonds and denaturing the protein in that egg, and that is cooking in essence. All right, that is cooking in essence the egg. Just like you were to heat it up and break those hydrogen bonds, you'd cook it that way as well. So There's more than one way to cook the cat, if you will. Okay, I think that I covered all. Oh, one more thing. So a dehydration reaction, right? All right, dehydration reaction is what we use to make the peptide bond. But to break the peptide bond, another type of chemical reaction is needed, and this is the chemical reaction we see catalyzed or made to occur more efficiently with the protease enzymes with digestion. We have to do what's called hydrolysis. Hydrolysis or hydrolytic, so hydro is water, lysis is lyse. So instead of pulling out a water molecule, what I need to do is I need to break a water molecule. Okay, so I have my H2O. I need to lyse that into an H and an OH, right? And so let's go back and just I'll give you my peptide bond here. Okay, something like this, right? So I have my I've got my peptide bond here. So what do I need to do? Well, I need to break that peptide bond. I need to lyse the water, right? And the hydrogen goes back to the nitrogen, the OH goes back to the carbon, and then that gives me that NH2, and then the COH can form again, and I've broken the peptide bond, and now I have my individual amino acids. So that's what we call a hydrolysis reaction or hydrolytic cleavage. And this hydrolysis reaction is not specific to proteins. We actually see this occur in other chemical reactions in the body, but it's certainly very important when it comes to breaking proteins down. Okie dokie, I think that we have covered all the relevant material we need to do. I do hope you guys find this to be helpful, and what we'll do is we'll see you in the next video. Take care.